Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 4.2, Introduction to Signal Transduction. Objectives today are to describe the components of a signal transduction pathway. We're going to be looking at reception, transduction, and response, as well as the different roles of each of these components in creating a signal transduction pathway. So we'll be looking at different kinds of receptors as well as different methods of transduction. And the suggested skill is Concept Application 1A, describe biological concepts and/or processes. So today we're going to start off by reviewing a concept from 4.1, and this is the signal transduction pathway, which, if you recall, contains three parts: reception, transduction, and response. Where a cellular signal is going to be received, it's going to be transferred to the nucleus, and then a response will be elicited. Now, the signaling molecule, or the ligand, is made to fit a very specific receptor. And this receptor is only going to be found on the target cell, the cell that the signal is going to. And this is because only the target cell is going to express the genes that are going to result in the creation of this receptor protein, the protein synthesis that will create this receptor protein. Because all cells have the same genetic information in them, in one organism. However, different cells express different genes within that set of information. And we'll cover this more in Unit 6. But these cell signals are going to be very specific because they're going to a specific target cell which contains a specific receptor. Now we're going to talk about the different types of receptors. And there are two large categories here of intracellular and cell surface receptors. And intracellular receptors are, as per their name, located inside of the cell, as you can see here in this diagram on the right. And they're going to be bound to by small hydrophobic ligands, known as steroid hormones. And the reason for this is that in order to bind to a receptor inside the cell, the ligand needs to be able to cross through the cell membrane, which, if you recall from Unit 2, is going to happen by small and hydrophobic molecules, because those that are polar, those that are large, they're going to need assistance in crossing that cell membrane. And these are going to directly affect the nucleus and gene expression because these receptors are already inside the cell. It makes it a lot easier for them to elicit a response. But of course, for other kinds of ligands, we need cell surface receptors. And this means there's going to be binding outside of the cell. And generally these receptors, and we'll look at some pictures on the next slide, have a portion of the receptor that is inside the cell that crosses the membrane that allows transmitting of the signal. And there are three kinds that we're going to look at. First of all, there are ligand-gated ion channels. And these are proteins that allow passage of ions across the cell membrane. And when the ligand is going to attach, the gate is going to open or close, allowing ions to pass through or stop passing through. And this is going to change the charges inside the cell, as well as, if they're hydrogen ions, the pH. Now, G protein coupled receptors, when the ligand is bound, they transmit a signal through the G protein. And there's going to be a binding of the GTP molecule. And of course, the ligand is going to activate all of this. And again, we'll look at a visual reference on the next slide. Now, the receptor tyrosine kinases are these enzyme-linked receptors that they're going to transfer phosphate groups to tyrosine, and this is going to be allowing to bind to growth factors. And of course, a kinase is going to be an enzyme that phosphorylates. It's going to add a phosphate group to the molecule, and this is how the signal is going to begin transmission. It's through this transfer of the phosphate group to the tyrosine. Here are some visual examples of our three cell surface receptors. Over here, we have the ligand-gated ion channel. As you can see, there's going to be ions on the outside of the cell, and we're going to have the receptor. And once the ligand, in this case a neurotransmitter, because these are going to be neurons apparently, the channel is going to open and these ions are going to allow to pass through because they are going to be traveling to create equilibrium. They are diffusing across the membrane, but of course they can only do so when the protein channel is open. Now over here we have the tyrosine receptor. And once signal molecules have been placed in both of the tyrosines, this is called dimerizing because there are going to be two of them. The tyrosines are going to become phosphorylated from ATP and will become activated, and this is going to activate the proteins attached to them, and a cellular response will happen. And this phosphorylation only occurs once the ligands have actually attached or bound to the tyrosine molecules, or the tyrosine receptor. And over here we have the G protein coupled receptor. This is the receptor, and we have our ligand that's bound to it. So this G protein is going to become activated. It's going to turn into GTP from GDP. It's going to become phosphorylated. And now this GTP is going to affect the effector protein, and the signal will begin to transfer downstream. So we've talked about the different kinds of receptors, both intracellular and on the cell surface. And we discussed how when the ligand binds to the receptor, there is going to be some sort of activation, some sort of response. But where does this signal now go? How does it transfer and actually create a cellular response? And this can happen through a couple of different ways, and the primary way for the signal transduction to occur is through a phosphorylation cascade. Now, just peeling apart the words here, we can see that phosphorylation is obviously going to involve phosphate groups, and a cascade means it's going to continue down. So binding of a ligand is going to change the intracellular domain of the receptor, allowing a pathway to begin that's going to relay the signal to its final destination, which is going to be creating a cellular response. Usually the signal is going to affect something in the nucleus, so we're going to have expression of certain genes. That will be a typical response. And the phosphorylation cascade is one way to activate the proteins in the chain through the addition of a phosphate group. And this is going to add a lot of negativity to each of the proteins that are going to be activated. So the transfer of the phosphate is catalyzed by the enzyme kinase, and then it'll later be removed by the enzyme phosphatase. And this is essentially just going to be amplifying the signal. So over here we have the signaling molecule, the ligand that's bound to the receptor. It's activated the relay molecule, and now the relay molecule needs to get to the cellular response. So how this signal is going to be transferred and even amplified is through the activation of protein kinases that are going to be phosphorylated. So you see the ATP is being used. It turns into ADP. We've got a phosphate group that's been added to the protein kinase, and as a result, we now have from an inactive protein kinase to an active protein kinase that is relaying the signal. And this is going to continue. It's moving on to the next protein kinase, to number 2, which is also being phosphorylated. And then the third one, number 3, also being phosphorylated. And once this signal has made its way through the phosphorylation cascade, our inactive protein has now become active through this transfer of phosphate groups that the kinases are relaying the signal through. And that's going to lead to a cellular response. And of course, phosphatase is going to then remove these phosphate groups from the kinases, turning them back to inactive protein kinases, allowing then another signal to be bound to the receptor and then be relayed to a cellular response again. And phosphorylation usually stimulates, but sometimes also deactivates a pathway that is already on. Now, phosphorylation cascades happen through proteins. Of course, each of the enzymes, the kinases that are activated by the phosphate groups are going to be proteins because they're enzymes. But there are small non-protein molecules that also can pass along the signal, and these are known as second messengers, because of course the first messenger is the ligand. And there are two main kinds here. First of all, calcium ions, and there aren't many in the cell, but a ligand-gated ion channel can allow calcium ions into the cell, and then proteins have binding sites for the ions, which will then change their shape, and that's going to result in the transfer of the signal. once the ligand-gated ion channel has been opened. Or more commonly, we'll see cyclic AMP or CAMP, which can activate a protein kinase A, a PKA, and that's going to phosphorylate its target. So oftentimes proteins or phosphorylation cascades or turning inactive proteins to active proteins is still going to occur even when you're using second messengers, but it's not just going to happen through a phosphorylation cascade. Cyclic AMP and calcium ions are going to help in this pathway. So over here we have the ligand that's going to bind to the receptor. And this is a pathway that uses cyclic AMP, and that's the signal is going to be transferred to the adenylyl cyclase, and ATP is going to be used to activate CAMP or cyclic AMP, and that's going to place itself in the protein kinase A, turning it into an active protein kinase, resulting in a cellular response. And what this enzyme does, adenylyl cyclase, converts ATP into cyclic AMP, allowing this protein kinase A to be activated. And of course, the PKA can have different targets, and each different target is going to create a different result. It's going to elicit a different cellular response. So phosphorylation cascade and second messengers are the two major ways for signal transduction to occur once the ligand has bound to the receptor and the signal is ready to be transmitted to create a cellular response. And there we go. That's the introduction to signal transduction pathways. Make sure you know intracellular and cell surface receptors such as ligand-gated ion channels, G-protein coupled receptors, and the tyrosine receptors, and also know phosphorylation cascades and second messengers. In 4.3, we will continue talking about the signal transduction pathway.